Okay, we're looking at membrane transport and different transport pathways. We've looked at diffusion. Special case of diffusion is osmosis. Remember, animal cells like to be in an environment where the net result of osmosis is zero because there's the same number of particles per volume in the cell as outside of the cell, so it's isotonic. In plants, the situation is different. Plants like to be in a hypotonic environment, meaning fewer particles around the plant cell. As a consequence, the plant cell takes out water by osmosis, and osmosis stops because of the pressure put against the cell wall. And that makes that plant is turgid. Okay, so if we have no simple diffusion, then we must have transport through a protein. And we can find out what type of protein might be involved by making a Michaelis-Menten plot. We call that a kinetics experiment. A Michaelis-Menten curve, this for you, we call it a kinetics experiment. So we can look for whether there is saturation at a certain substrate concentration, and we can also investigate if there's a need for energy, for instance in the form of ATP. And so we can distinguish by the type of protein that might be involved. Is there saturation, yes or no? If there is, then we think about carriers or pumps, we'll see later. If there's not, then we think about channels. The special case for transport are electron transport chains that function in respiration and photosynthesis, and that we will see a little bit later in section 2. Or we can distinguish whether the protein needs a source of energy such as ATP. If it doesn't, then we speak of passive transport. If it does, then we speak of active transport. So active transport requires chemical energy, metabolic energy, which is then used to transport other molecules from an environment with a low concentration into an environment with a high concentration. Those are unfavored reactions. So let's have a look. And we can look at our Michaelis-Menten plots. And if we get a curve like this, a very steep straight line, then, and we never see saturation, then we're suspecting that we're dealing with a channel or a pore, a protein that forms a hole in such a way that there's never a need to stably bind to the substrate, it just moves through. So this idea, a molecule moves straight through from a high to a low concentration. Famous example in animal cells are the sodium channels. We have sodium channels in our membranes, and in the open state, these channels allow the quick flux of sodium ions from the extracellular environment into the cell. Now, if we would have our channel in another state, maybe we get no transport at all. It's just flat line. That means the channel is closed. Many channels are what we call gated. That means they can be closed or open. Pores are also sort of channels, but they're actually typically not gated. So channels or pores, holes in the membrane formed by protein channels, often are gated. We can have an open and a closed state. So we can think of channels in a simple diagram like closed or open. And the opening-closing mechanism might be controlled by different factors depending on what protein we're looking at. Some channels are gated by charges, by membrane potentials, as we will later see. We call those voltage gated. Some are gated by the binding of other molecules. We call those ligand gated. And some are gated by mechanical force. So they're sensitive to touch. So we can have channels in a closed state and an open state. In the open state, there's rapid quick transport. We can have pores. Pores are found in the outer membranes of bacteria, gram-negative bacteria, and also in the outer membranes of mitochondria and the outer membranes of chloroplasts. And these molecules have this structure where they have beta strands together that form the pore structure. Here's a diagram of such a pore called porin. And it allows for the quick flux of small molecules through this space. Now, the channels, they might be selective. They might be selective for the charge of the particle and the size of the particle that goes through. And important selective channels are the potassium channels and the sodium channels that we have in our cell membranes. They're very important for communication between cells, especially communication in the nervous system. So the whole action of neurons, that all depends on the channels. So we'll see that a little bit later in the class, how that works. So channels, they don't stably bind the substrate, but they have some selectivity. So how can you imagine that channels are selective? We can just have a pore, a hole, that has a certain size, meaning that large particles are excluded. And this is really how we think the porins work. They exclude things that are larger than 3,000 times the mass of a hydrogen atom. So 3,000 Daltons doesn't pass through a porin. But that still means that very small peptides can pass through, and that most amino acids pass, or all amino acids actually, nucleotides, so a lot of things pass through porins. You can also have selectivity by charge. You can imagine that if you have a pretty narrow opening somewhere in your channel, and if there are a lot of negatively charged amino acids that line that channel, so glutamate and aspartate, then by repulsion, negatively charged ions don't want to go through, but positively charged ions can flow through such a channel. So you can have charged selectivity. And if you get a combination of both charge selectivity and size selectivity, then you can actually get very specific channels. So specific for potassium versus sodium, which is different in size. Okay, so we can have selective channels and they can be gated on top. Special case of channels are channels that allow the rapid movement of water. I've told you osmosis, diffusion through a membrane without using a protein is, if we think about the individual water molecules, something that's not likely. But there's a lot of water, so the net rate of osmosis is always appreciable. But there are cases where fast water flux is desired. And in that case, there are special channels that we call aquariums. These have been discovered maybe 15, 20 years ago. Fairly recent discovery, at least for me, for my lifetime. So here I have a picture of an aquarium, and, well, let's make it dark. Somewhat dark. So this structure is really based on a model of how we think the protein sits in the membrane. It's not maybe directly an X-ray structure that it's based on. And you can see here in red and white, those are the water molecules. So red is oxygen, white is the hydrogen. So it's really a chain of water that can move through. This allows for a much more rapid diffusion of water through the membrane than you can have without such a water channel. Now we know that these water channels allow for fast water transport based on some interesting experiments. So what we're looking at here at the bottom is, you can think of a phase contrast light, or definitely some form of light microscopy, where we're looking at two oocytes. This is sort of a Novarski microscope that was used here. And we're looking at a time lapse. So oocytes are pretty large cells, especially if we go for oocytes from frogs This can be visualized pretty easily with a microscope, a light microscope. I'm going to give you some more light. Because you've had your lunch. This will wake you up. So, how do we know about the function of aquariums and that they give this rapid water transport? And we have two cells. Both cells we stuck a needle in. And in one needle we had some control RNA that didn't encode for this protein and encoded for something else or for nothing. But that cell we did stick it with a needle. And the top cell, we put a needle in, and in that solution in the needle that we stuck in with the syringe was some mRNA encoding an aquarium protein. So that mRNA got translated, the protein got formed, and it inserts in the membrane of this cell. And then we start to put these cells in a solution that has less salt. So a hypotonic solution. So at time 0, not shown here, both cells were placed in a hypotonic solution with lower salt concentration. So in both cases, water by osmosis wants to move into the cell where there's a higher solute concentration. But what we see is that in the top cell, it's swelling much quicker. This is a time-lapse series. And ultimately, that cell starts to burst because of all the water it's taking up. So that shows us that osmosis is quicker in the top cell that has the aquarium expressed. Got it? So it has to be a light microscope approach here. The cells are alive, some form of normality that we're looking at. The water channel, the aquarium, recently discovered. Okay, now, so we can have channels. What if we get a Michaelis-Menten plot and we get this nice saturation type curve, which we know so well from our enzymes? So in this case, we have the rate of transport is defined by this equation where we really have a dependence on the concentration, and we have a Vmax and we have a Km, very similar to enzymes. This is really just the Michaelis-Menten equation where the C here stands for the concentration that the transporter is seeing for its substrate. So we have a saturation because we have a protein that binds the substrate, and the more substrate we give, at some point we start to occupy all the binding sites, just as in an enzyme. So carriers are therefore also specific. This type of kinetics, we call that the kinetics for a carrier. Carriers are specific. They bind a specific molecule with stereospecificity and a lot of precision. So the transport will depend on the concentration. This transport depends on the concentration, as we can see, and it follows the saturation Michaelis-Menten kinetics. So, for instance, a molecule like glucose, it cannot go through the membrane by diffusion because all of the hydroxyl groups on glucose, they want to be hydrogen bonded with the surrounding water. So water doesn't want to let go of that glucose. But if we have here a protein in the membrane and that protein has a space to bind the glucose, and if we think of a very subtle conformational shift in that transporter or carrier protein, then we can envision that the substrate molecule can first enter from one side and then be released on the other side. We call it a flip-flop mechanism. The type of conformational change that we see here in this transporter, in this diagram, is certainly exaggerated. The conformational changes are very, very subtle in reality. So the substrate can be released and then the protein can flip back and it can take another molecule. Now, every reaction is reversible, so everything here can go in the backward or in the forward direction. the net transport direction depends on the concentration difference. The molecule, like glucose, will move from the higher to the lower concentration if you think about the net direction of transport. But there's some transport back, but there's faster transport forward. There's fast transport from high to low and slow transport from low to high concentration. Think about it like that. So we can say there's a concentration gradient, and I'll use that term a lot. What I mean with that, there's a difference in concentration for a molecule on one and the other side of the membrane. So the net transport is down gradient, however it occurs in both directions, remember. The system is typically very specific to the substrate of stereospecificity. So if we have D-glucose normally in living organisms, then the L form would not be transported by this transporter. And because there's a binding pocket, we might have inhibitors, competitive inhibitors. Glucose is very similar to another molecule, galactose, another sugar. Galactose might be transported by this transporter very slowly, and it might competitively inhibit the transport of glucose. Or if we have modified versions of glucose, where glucose is chemically modified, then such a molecule might act as an inhibitor competitively of this transport pathway. So carriers, or transporters as we call them, they take a molecule and allow it to get over the membrane by what we call a flip-flop mechanism. It involves binding, goes down gradient. Okay, now this was one possible version of the carrier, and we call that the uniporter. So a uniporter takes one type of molecule, and it moves it over the membrane down gradient. So in this diagram here, this little triangle, this little triangle here depicts a difference in concentration. So we're having a high concentration on the top here for our black molecule, let's say glucose, and a low concentration on the bottom. And the transporter, which uniporter, moves the molecules in the net direction down gradient. But there are other options. Many carriers take two substrates at the same time. And if they do that, they can move the two substrates in the same direction or in the opposite direction. Same direction or in the opposite direction. If they move in the same direction, we speak of symporters. So imagine you have here one molecule, and it has a concentration gradient that allows it to move down gradient here to the bottom, to my little red molecule. We can now couple the transport of that red molecule, for which there's a favorable concentration difference, with the transport of something else that then actually can move up gradient. As long as the gradient that drives the process is stronger than the gradient for the second molecule. So that's symport. And a very similar idea, we can have antiport. So one molecule moves down gradient, and that allows another molecule to move up gradient, but this time in the other direction. So the idea is this all depends on subtle conformational changes in the carriers. Now it turns out that cotransporters are really important, and they're found in most cell membranes and also in a lot of organelle membranes. And the molecules that move down gradient in order to facilitate the uptake of something else are almost always one of these three: protons, hydrogen ions, which is really the most common case; sodium ions, which is very common if you think about the plasma membranes of animal cells; or chlorine ions. There can be other driving gradients, Potassium is a possibility, but these are the three major ones, with really protons and sodium ions being the most important. So for instance, we use sodium gradients. We use the fact that outside of our cell membranes there's a lot of sodium, inside there's a low sodium concentration. We use that sodium gradient to drive the uptake of many food substances that we need, like sugars and amino acids. We get sugars and amino acids into our cells by these Symport mechanisms, typically using sodium for us to get it into the cell. And you can ask the question why this is the case, so let's answer that question a little bit later. So it's the case. So Symport, antiport, uniport, all forms of transporters or carriers. Okay, so it now means that we can use carriers, symporters, to move molecules against a concentration gradient. So in that case, the transport is always coupled to the down gradient transport of something else. That other molecule I call as the driving gradient. So sodium for the mammalian plasma membrane constitutes the driving gradient is composed of sodium. High sodium out, low sodium. So that now means we can actually move a molecule into a compartment with a higher concentration, which is energetically unfavored. It's energetically not favored. It actually means that the energy stored in that second molecule goes up. So it's actually a form of active transport, but since it depends on a gradient formed by another molecule, we'll see a pump, we call this secondary active transport. So here's an example, that's just a picture from the book. It depicts how leucine can be taken up in our cells together with sodium. So there's a sodium gradient. That sodium gradient was made by the cell by other mechanisms. There was an investment of energy there. And now leucine can be taken up by binding together to the carrier with the sodium, and then it can be released on the other side and go back. We call this secondary active transport. Keep in mind always that all the steps here are in principle reversible, and if the gradients are reversed, then the direction of transport actually reverses. Okay, so we call this co-transport. So we have carriers. In the carriers we have uniporters, one molecule. We have co-transporters. Co-transporters are the symporters and the antiporters. Okay, third possible mechanism. Again, we might have a saturation curve indicating there's a specific binding site for the molecule that gets moved across the membrane. Saturation is specific, but in this case, it also uses ATP. If that's the case, then we think of a molecule that we call a pump. Pumps are membrane proteins that use the energy stored in ATP to drive a molecule across the membrane. So they're sectionable, they follow Michael's mental kinetics. There's again the equation for that. There are, in biology, different kinds of pumps, and there are three major and important types. The two most important ones are indicated here in red. So the three major classes we see in nature are the so-called P-type or P-class pumps, where P stands for phosphorylated intermediate. These are molecules, enzymes, that phosphorylate themselves, that causes a conformational shift in the protein, and it functions as a pump. We have the V-type pumps that are commonly found in vacuoles, for instance in fungi, and also in plants. And some of those use the energy stored in pyrophosphate. Phosphate coupled to phosphate. It's a high energy bond. If you break that, there's energy released. V class or V-type pumps use that energy. Medically important are the so-called ADC pumps. ADC stands for ATP binding cassette pumps. They're also known as the multi-drug resistance proteins, or MDR proteins. There are two types of contacts where this transporter is medically important. First of all, the ABC transporters, how they work, is they bind ATP, hydrolyze ATP to drive a variety of substrates across membranes. There's a lot of diversity within the ABC type of pumps. In bacteria, the ABC pumps are important in pumping out, removing from the cell toxins, such as antibiotics that we might want to throw at bacteria that are causing a nasty infection. Turns out that many of the antibiotics that we come up with stop working. Why is that? Because there always is an organism out there in nature that has a gene sequence encoding for an ABC pump that can remove that antibiotic from that cell, making the cell resistant. Therefore, multi-drug resistance. Similarly, in tumors, we also have sometimes problems with this type of pump. What's going on there? We're trying to treat the tumor, the cancer, with a drug killing those cells. So if I have drugs that target rapidly growing cells, they preferentially target tumor cells because they grow rapidly. So many of those drugs become inactive in cancers because in cancers sometimes the control over the genetic system is lost and you get extra high expression of naturally present pumps that serve to remove especially hydrophobic molecules from a cell, causing again resistance of the tumor. So ABC pumps are important medically because they often are a mechanism by which the problematic cells, whether it is the microbe or the tumor, becomes resistant to whatever drug you will treat with, multi-drug resistance proteins. P-type pumps are very important in nature as well. They're important molecules especially to set up ion gradients. In our neurons, 70% or so of the ATP is consumed by a P-class or P-type pump. Okay, so the P-type pump is very well studied, and here's just a picture from the book of what we think this molecule looks like. It's a model. The top here is based on an X-ray structure, the bottom is based on an X-ray structure. What's in the membrane is based on other experiments, not X-ray structure. So it's a model based on combined evidence. We see here it binds somewhere ATP, and the rubidium ions here, that mimics potassium actually, rubidium potassium. It's often for a protein seen as the same. So this is what the P-type pump looks like. How does it work? The P-type pumps are pretty important. Here is the major pump, which is called the sodium-potassium pump, which maintains the main sodium gradient across mammalian cells. And it consumes 30% of the energy in most of your cells and up to 70% of the energy in your neurons. So it's a lot. How does it work? Well, the enzyme, it is an enzyme. It sits in the membrane. It has two conformations that we call E1 and E2. In the E1 conformation, the protein can bind sodium from the inside of the cell. There's some sodium there, a couple of millimolar. They can bind here to the protein in a binding pocket. The protein can then bind ATP and it can phosphorylate itself. So it's a kinase, and it is an ATPase at the same time, and it uses that ATP to put a phosphate on itself, and that drives the conformational change, going from the E1 to the E2 state. So we're now phosphorylated, go to the E2 state, and now that binding pocket for sodium is exposed on the exoplasmic side. So sodium can move out, and it was free sodium that binds. Potassium can now bind, and it's 2 potassium that can bind. When the potassium has bound, the molecule can ultimately become dephosphorylated, and then it flips back to the E1 conformation. The potassium can flow out, and the molecule can be binding ATP again. So just to summarize this, there are 2 conformations, and what happens in a cycle of ATP binding and hydrolysis, for every 1 ATP that is hydrolyzed, there are free sodium removed from the cell, and in exchange 2 potassium are allowed to come back in. And I think you should know the numbers there. They are really important later. So free sodium are pumped out, 2 potassium pumped in for every ATP that's hydrolyzed. And you can ask the question, why is it free sodium, 2 potassium, and not something else? I can give you part of the answer. I can give you a much better answer in a week or so. A little bit over a week. So why sodium for potassium? Well, if we move sodium out of the cell, we're not just moving an ion against the concentration gradient, we're also moving charges. And as we move more and more charges, an electrical charge difference starts to build up. If it becomes so big, it becomes impossible to move more sodium out. Therefore, it is necessary to move some potassium in, because that potassium compensates, at least in part, for the charge. And it all has to do with the amount of energy that is stored in 1 ATP, which allows